Hello, my name is Christian Nogales. I'm a postdoctoral researcher in the Department of Pharmacology and Personalized Medicine at Maastricht University. Over the last 5.5 years, I've been heavily involved in the REPO trial project, where my focus has been applied bioinformatics and diagnostics. And that is what I would like to walk you through today. So let's get started. If we look at current disease definitions, we realize that they follow an outdated last-century organization, where diseases are defined by the organ they appear to affect the most, such as stroke in the brain or COPD in the lungs, one or more symptoms, such as hypertension, or to make matters worse, the name of the doctor that first described the disease, such as Alzheimer's, Parkinson's, or Crohn's disease. This has fundamental consequences by the way we approach, diagnose, and treat diseases, leading to pharmacological treatments that are symptomatic and quite ineffective, resulting in high numbers needed to treat. Therefore, we need a new way to approach diseases, but also to diagnose and treat them. In 2007, the first comprehensive human disease network was introduced. In this network, diseases are represented by nodes or circles and connected to each other if they share a rich genetic association. Consequently, we can see how diseases interact with each other, and more importantly, obtain a better view of their causal genetic origin. Importantly, the genes connected to these diseases are the potential lead toward new disease definitions and disease modules. But what are these disease modules or networks, and how do we define them? Basically, it has been observed that in a protein-protein interaction network, proteins related to the disease tend to interact closely to each other, creating disease modules. To build these disease modules, we can incorporate the genetic information from the human disease networks, as well as other disease information, such as comorbidities. Those genes can be used as seeds and will be the starting point to build our disease module. They are then mapped to the human protein-protein interactome, where we explore their neighborhood, eventually extracting the disease module and disease mechanism. In the original version of the disisome, a heterogeneous cluster of cardiovascular and metabolic disease phenotypes appear, including diseases such as diabetes, stroke, asthma, hypertension, but also Alzheimer's and Parkinson. The fact that they appear together linked by shared risk genes suggested that they might share a common underlying pathomechanism. Indeed, several genes related to reactive oxygen species dysfunction and CYP signaling, initially separated fields, appear to link these phenotypes in one joint signaling network, ideally suited as a test case for the mechanistic redefinition of diseases. Therefore, to start building a disease module, we started exclusively from clinically validated ROS and CYP-related seed proteins. Those were mapped to the human protein-protein interactome to start a ROS CYP disease-relevant module. Here we compare two different local methods that yielded similar results. A first neighbor method that controls for highly connected proteins, and the Diamond algorithm that identifies disease modules according to the connectivity significance of the proteins in the module. Both methods identified an OX4, 5, NOS STC intervention module, ideally suited for an pharmacology therapeutic approach. Nevertheless, not all patients with one of the disease phenotypes have developed the disease through the same mechanism. The challenge is thus to develop diagnostic strategies to detect those patients and only those patients that have this causal disease mechanism. We could then select and treat these patients, ideally reducing the number needed to treat to 1. Or in other words, all patients will benefit from the therapeutic intervention. With this purpose, we developed a diagnostic assay where we endotype patients from the ROS CYP disease cluster. This assay combines ROS-related markers and CYP-related phosphomarkers. NOS5 levels and hydration markers induced by ROS, like netatyrosine, are stable enough to be measured in stored plasma or serum samples from biobanks. However, on the other side, protein phosphorylation induced by CYP is not stable enough to be measured in stored samples. This way, the phosphorylation is induced in vitro, and the phosphoproteins are measured directly after the stimulation. Like this, we can stimulate different parts of the pathway, like for example, with HSC stimulators and activators. And by comparing the response to healthy controls, we can identify the ROS CYP dysfunction. To give you some examples, we collected blood samples from ischemic stroke patients in different biobanks from Barcelona and Essen, and time points after stroke, and we measured metatarosine. We saw the highest levels of metatarosine appear from 4 to 8 hours after the event, suggesting this time frame as the best time window for our treatment. In addition, we also see an increase at later time points, probably associated to inflammation, which is not the target of a mechanistic therapy. Another biomarker of the ROS-CYP endotype will be the ROS-forming enzyme NOX5, which can easily be detected in circulating microparticles and plasma. Here, in hypertension, one of the diseases of the cluster, we were able to detect a subgroup of hypertensive patients with high levels of NOX5 compared to healthy individuals and other hypertensive patients, which we hypothesize have developed hypertension through the ROS-CYP mechanism described and are potential candidates to another pharmacology combination targeting the causal mechanism. The same thing we can observe here in heart failure with preserved ejection fraction, or also known as HFpEF patients, where patients with plasma NOX5 levels higher than the determined cutoff are considered to have developed the disease to the ROS-CYP endotype. In order to fully detect the ROS-CYP endotype, we also develop a cell space assay. Since more than 65% of all proteins are produced in most cells and tissues, we believe that we can analyze more than 65% of signaling pathways present in any cell or tissue. This means that for heart kidney-related phenotype, the mechanistic dysfunction can be detected in other types of cells not located in the heart or the kidney, such as blood cells. Therefore, to develop our diagnostic assay, we use blood samples. Moreover, while designing the assay, we had two main goals. One, to have a protocol that is simple, but also fast in order to be easily implemented in a clinical setting, where maybe only basic lab equipment is available. So from one blood tube, we select a mix of cells after a one-step fast centrifugation. These cells are preserved in a way that ensures sufficient cell viability. The signaling pathways can be investigated later on with a cell-based stimulant assay, where cells are stimulated with drugs and downstream signaling responses are measured. For example, to detect the ROS-CYP endotype, ApoSGC can be probed using SGC activators, and SGC using SGC stimulators, and the downstream phosphorylated BUS response to the stimulant is used as the final readout. Finally, in preparation for the RIPPO-HFpEF-2 trial, we tested the two biomarkers, and here we show how they can be used to detect the ROS-CYP endotype in HFpEF patients. NOX5 measurements were performed in plasma samples, and phospho-BUS measurements were determined from a cell-based assay. Altogether, we see that NOX5 plasma levels and the SGC activator stimulator need to be evaluated, since these biomarkers rarely overlap. Therefore, we show that both biomarkers are necessary to identify all ROS-CYP module pathomechanisms. Finally, I would like to thank you for your attention. And of course, feel free to reach out to us. We're happy to discuss, to further engage in further discussion. Thank you so much.